Hello students, welcome to EPG Pathshala. I am Dr. Vikrant Nain from Gautam Buddha University, Greater Noida. Today we are going to talk on module Identification of Drug Targets and Screening of Inhibitors, Modulators under the paper Molecular Therapeutics. Our learning objectives are drug discovery process, selection of drug targets, screening strategies, and computer-aided designing. Drug development. Drugs are used for enhancing the quality of the human life as well as increasing the length of the human life. But the different organizations develops drugs for the different purpose. Like government organizations, they have the social responsibility, but at the same time the business houses, they are, in addition to the social responsibility, the profit making is also a important factor while deciding the which disease to be selected and against which the drugs need to be developed. Till now, the infectious disease dominates over the other diseases, particularly into the developing countries. How we define disease and drugs? Disease is a condition of a person, animal or plant in which its body or structure is harmed because an organ or its part is unable to work as it usually works. Similarly, drug is any chemical that is used as a medicine to cure a particular disease. Drug molecules targets some specific molecules either inside the cell or on the surface of the cells. On the surface of the cells, there are the receptors that are present, the ion channels are present, and inside the cell, the different biochemical processes are going on. Different drugs targets either the receptor and change the signaling process, they targets the ion channels and change the permeability of the membrane to the different molecules, or they inhibits the different enzymes or the transcription factors inside the cell, particularly the enzymes, and they inhibit a target pathway. Classification of the drug targets. Drugs can bind to another molecules in the cell. Those molecules may be lipids, generally they are present in the cell membrane or the cell membrane lipids. There may be proteins. Proteins can be further classified on the basis of their function. There may be receptors present on the cell surface. There may be enzymes catalyzing different metabolic pathways. There may be the carrier proteins, again present on the cell membrane, or there may be the structural proteins providing the particular shape and size to the strength to the cell or the tissue or an organ, and there may be the transcription factors that can go to the nucleus and regulate the expression of the another genes or the production of the another type of the proteins in the different categories or in these receptor, enzyme, carriers or the structures. Further, the targets may be the nucleic acids. Among the nucleic acids, it may be DNA or RNA. This RNA may be protein coding or non-coding or the regulatory RNA. Sometimes these carbohydrates generally present on the cell surface can also be targeted as a drug target. Receptors and the enzymes are the major class of the molecules that are used for the drug development. Hormones and factors, nuclear receptors, ion channels are also used, but with the less frequency. And if we say that every cell is made up of the different type of the molecules like carbohydrates, lipids, proteins, but these are the proteins that figure out as the dominant target molecules, whether it is a receptor, whether it is an ion channel, or whether it is an enzyme, or it is a transcription factor. These are the proteins that are the major class of the molecules that are used for the drug development. To design a drug, first we require a drug target against which a drug can be designed. Then what should be the characteristics of a good drug target? First important characteristic of the drug target should be selectivity. It's targeting, if we are targeting a bacterial enzyme which is not present in the mammals or the host in which that particular enzyme infects, or which has significant structural differences from the corresponding enzyme in the mammal. Second one is specificity. Specificity is that targeting one subtype of a receptor or an enzyme may enable the pharmaceutical chemists to avoid potential troublesome side effects. Less variability in the population. Another characteristics of a drug target. Suppose the drug target is hypervariable, then a drug molecule may be able to bind to the certain part of the population but not the whole population. That means drug targets should be less variable in the population. Then the chosen target may overcome time lose its sensitivity to drugs. That means the drug targets should not get easily the variations in that. That means the drug should be highly specific, and that's possible only when the target is not hypervariable and is not easily mutated with due course of the time. So when we are selecting a target, suppose there are different strategies when we are selecting to cure a disease, that is because of the pathogen. In that case, the selecting target is done in a way that the target is generally either it is present only in the pathogen or and absent in the host. And the identifying target which are shared but which are significantly different structure. The basic philosophy behind this is the drug molecule should specifically bind to the targets present in the pathogen but not in the host. Either it should not be present in the host, or if that particular enzyme or the target is present in the host, that should be structurally variable so that the pathogen protein can be specifically targeted. When we are selecting a target within an organism, the difference between the different enzyme and the receptor is taken into consideration, or the difference between the receptor types and the subtypes is taken into the consideration, or sometimes the difference between the isoenzymes is taken into account, and sometimes the expression of the target in the different organs and the tissue is taken into account. So, for this drug discovery process, it starts with the target identification and then the target validation. How to identify a drug target? As we have discussed just in the previous slides, that first we need to identify a target. That if we are selecting a target between the pathogen and the host, then we can use the genomics, proteomics, or the microarray or the transcriptomics approaches. For that, this differential expression or differential or the presence of the specific targets in the pathogens and the host, they are identified. With the genomics approaches, we sequence the whole genomes, and using these bioinformatic tools, we identify which particular pathways are present in the pathogen but absent into the host. For example, hemoglobin degradation pathway. It is present in the plasmodium. It is present in the plasmodium, but not into the host. So that pathway is generally targeted for the development of the antimalarial drugs. Similarly, proteomic approaches. In the proteomic approaches, we identify the proteins that are present into the pathogen but not into the host. And such proteins after characterization may be targeted. Another approach of the microarray and the transcriptomics. These two approaches are to study the genes expression patterns, and the differential expression of the genes into the different tissues or the different organs can be first lead to the identification of the targets and later on validated, and those validated targets can be used for the drug developments. After identification of the target, it is the how to validate the target. For the target validation, there are the common strategies, the gene knockout. That means after knocking out of the gene, the organism should not be able to survive, or it should prove the essentiality of the target gene to that particular organism or the function of that particular tissue. For that, either knockout or the enzyme inhibition assays are carried out. Once we ensure the essentiality of that particular enzyme or that particular gene for that particular trait or that particular disorder, then we can select it as the drug target and then move to the identification of the drug molecules for that. For that, first step is the lead identification, and later on we go for the lead optimization. For the lead identification, we go for the high throughput screening. That screening may be enzymatic screening or this virtual screening. Enzymatic screening means we go for the direct enzyme assay. We take the large library of the compounds, and in the enzyme inhibition assays or any other binding assays or the receptor activity assay, we identify the lead compounds in the direct assays. But in case of virtual screening, we first computationally screen the library, and then from those screened compounds that are selected from the library, from a virtual screening or the computational screening, we select them for the assays, and the enzymes or the compounds that lead to the inhibition of the target enzymes are selected. But generally, these selected compounds are either not that much specific or their activity is not sufficient. For that, we go for the lead optimization. For the lead optimization, we use different strategies of this medicinal chemistry. We go for the different studies, quantitative structure activity relationship, ADME profiling, or this bioavailability assays. Once we have optimized our lead, we go for the clinical trials. The first, we go for the preclinical assays. For that, in that we study the thermodynamics and the pharmacokinetics. Pharmacodynamics is what drugs do to the body, and pharmacokinetics is what the body do to the drug. And this ADME, that is absorption and metabolism, excretion and distribution into the body, and then the toxicity testing through the animals. Once these pharmacodynamics and the pharmacokinetics assays are done, then we go for the toxicity testing over the animals, and the molecules that prove safe to the animal test are taken to the clinical phase or the human trials. The drug discovery process. The drug discovery process starts with the selection of the disease, which disease we want to target. Once we select a particular disease, then we go for the identification of the target molecule inside, whether into that host or in the pathogen that will be targeted with the drug. Once we select the target and characterize and validate that it is a valid drug target, we go for the identification of the molecules that can be used for the binding to that particular target. And this process is called hit identification. Hit identification defines only the molecules that can bind to the target, but it doesn't mean they will be the functional drugs. Out of those hits, they are used for the different assays. We go for the lead identification. Then, to increase the potency of the lead molecules, we go for the lead optimizations, and then for the different preclinical and the clinical trials before the release of that particular drug. For drug development, there are the two major class of the strategies that are used: target-based reverse chemical genetics or original drug design, and phenotype-based forward chemical genetics. The difference between these two steps. The target-based reverse chemical genetics approach starts with the selection of a drug target and characterization of the target molecule. And once you select that particular target molecule, we go for the screening of the different chemical compounds that can bind in and inhibit or activate the target. And once after the screening process, we find the lead compounds. Those lead compounds are tested in the clinical, preclinical and the clinical trials. While in the phenotype-based forward chemical genetic approach, that compounds are directly tested on the disease model organism. And the compounds that are identified to be effective are later on validated to identify their targets. For the target identification, we can take comparative genomics based approach. In the comparative genomics based approach, we compare the target genomes of the host as well as the pathogens. And then through the computational biology, we identify the pathways that are present in the pathogen but then not into the host. And then in those pathways, we identify the genes that are essential for the functioning or survival of that particular or pathogen. And such targets are selected for the drug designing. Once the targets are identified, then validation of the essentiality of the target in the organism of interest is essential. For that, these knockout studies are generally taken off. Nowadays, this siRNA and the CRISPR-Cas9 based approaches can be easily adopted for that. Once a target is selected and validated, after that, we go for the cloning, heterologous expression and purification of the target protein. And because that protein, the target protein, is required in high quantity for the further assays, and that can only be produced by the heterologous expression in the hosts like E. coli or another host, yeast, but generally most of the experiments are carried out with E. coli, and for the heterologous expression, cloning of the gene is essential. In these experiments, sometimes that the expressed protein is not functional, so for that, enzyme assay with the heterologously expressed and purified proteins are essential. Once these functional assays are done with the recombinant protein, then these enzyme assays can be used for the testing of the drug molecules that have been selected either through the virtual screening or the hydrophobed chemical screening. With the emergence of the genomics era, where the large number of the genomes are getting sequenced day by day, and the genomes of the different pathogens and the human and the other organisms are available. Not only the genome sequences are available, the computational tools are available that can predict the presence of the particular genes. How many genes are present, where they are located. These gene sequences can be translated to the potential proteins, and the function of those proteins can be predicted through the comparison of those protein sequences with the sequences of the another organism where that particular gene have been characterized. This in annotation and the functional characterization or functional prediction of the different these sequences provides an opportunity to identify the different drug targets. Like suppose we want to identify a target for a drug target for a particular pathogen. All the proteins of that pathogen can be compared with all the proteins of the human, and the proteins that are specifically present into the pathogen can be used as a drug target. How this hemoglobin degradation pathway that is particularly present into the plasmodium but that is not present into the humans can be used for the making anti-malarial drugs. All the steps of this hemoglobin degradation have become a valid targets for the anti-malarial drug development. And among that, for an example, this PFA-M1 and M17 that are the neutral aminopeptidases are being pursued for the development of the anti-malarial drugs. It is not only the presence or absence of the particular gene that can be used as a drug target, it is the expression of the gene, and the expression also sometimes varies quantitatively. Specifically into the non-pathogenic diseases, the expression of the different genes, or quantitative expression of the different genes is responsible for the different disease development. Like the example, this HER2 breast cancer, it is because of the overexpression of the HER2 protein into the cells. And these techniques of the microarray or transcriptomics can be used for the comparative functional genomics for the identification of the drug targets for the known pathogenic diseases. Druggable target. Can every target be used as a drug target? No. Why? First, we have to ask a question: does it have the sequence or domain similar to the known targets? Second, does the target have a site where drug can bind and with appropriate affinity? Because it is essential for a drug to specifically and appropriately with appropriate affinity. With appropriate affinity means that if the binding affinity is less, then the more dose of the drug will be required for the desired effects. If binding affinity is high, then at the low concentration also the drug can bind to the target and give the desired effects. So, similarity with the other domains. In the proteins, it is not only the proteins sequence that is required to be diverse, it is the three-dimensional structure of the domain that is targeted for the drug. It should not show very high similarity with the another domains, because if there is a similarity, then there is the possibility that drug can bind to the another proteins also and show the off-target activity. Once drug target is identified and characterized, next step is cloning and expression and the development of the assay for that particular gene. For this purpose, heterologous expression of the protein is done. During the heterologous expression, we specifically into the E. coli, and if it is not in the E. coli, sometimes then other organisms like the yeast or some other system are used. Once these proteins are overexpressed and purified, those proteins can be used for the assay of the drug molecules and to validate the drug molecules that are identified. So, for any drug identification process, the availability of the compound library is essential. This library may be in the compounds of the natural sources. It may be that we create the large number of the library of the compounds on the basis of the existing compounds and make and then increase the size of the library through the combinatorial chemistry and make the derivatives of the already known compounds. And from that library, we can identify the synthetic chemicals that have the much more potency and efficacy than these natural compounds. And these pharmaceutical compounds has tremendously invested in the development of these compound libraries that can be used for the drug discovery process. Once a drug target is identified, it is characterized and available for the assays, next step is identification of the lead molecules. With the availability of the large number of the natural compounds, synthetic compounds, and another similar compounds that are chemically synthesized, large library of the compounds are nowadays available. Out of this large number of the compounds or these millions of the compounds, how the effective molecule can be identified. For that purpose, two strategies are used: high throughput screening and the high throughput virtual screening. In the high throughput virtual screening process, the chemical compounds are directly used for the assay in a high throughput manner using the robotic workstations. This process gives in the directly the compounds that the effective compound that can be used. But the limitation of this process is high cost, because we have to get the millions of the compounds for the assay and the cost of the assaying also. To overcome this process, the high throughput virtual screening approach can be used. In the high throughput virtual screening approach, the 3D structure of the target protein is taken and the cavities are mapped over the surface. Those cavities are generally the active sites or the allosteric sites. And the computationally, these library of the compounds are screened, and the compounds that effectively docks into the active site or the selected allosteric site are selected as the lead compounds. For high throughput virtual screening, two approaches are used: the ligand-based design and the structure-based design. The ligand-based design is used when the 3D structure of the target molecule is not known. And when the 3D structure of the target molecule is known, then this structure-based screening is used. In the drug discovery process, the application of the computers or in silico screening of the compound library or the high throughput virtual screening has increased tremendously. The reason for that is that available with the large number of the genomes, the sequences of the large number of the genomes, the number of the targets that can be used as the drug target have increased. Further, not only it is the nucleic acid and the protein sequences that are available, with the availability of the expertise in the crystallization of the proteins, the 3D structure of the druggable targets have increased. 3D structure of the proteins have increased. Availability of the 3D structure of the proteins have increased. And further, this computational power, it is gradually increasing. Communication technology, it is increasing the communication between the different databases and the users, and that have led to the availability of the large number of the libraries, computational power, softwares that can be used for the computational screening or computational drug designing. This computer-aided drug designing is more suitable when the structure of the target is available. And then that structure can be used for identification of the drug molecules that fits in the cavity or the active site of the protein and can use as an activator or an inhibitor. But the drawback of this is that only a limited conformations are sampled. That means, first, in the 3D structures that are available, they are static in nature. But in nature, proteins are dynamic. They are continuously changing its conformation. Similarly, the drug molecules also, they can adopt different conformations. And to sample all those conformations is very, very difficult and time consuming. So only a limited conformations are sampled, but that lead to the inaccuracies into the drug discovery process. Or many compounds that are selected as the inhibitors, later on they may not be found to be the potent inhibitors because of the sampling limitations. With an example of the PFA-M17, the high throughput virtual screening protocol is described. Here, the 3D structure of the PFA-M17 is taken. That has been already resolved by the X-ray crystallography. The region that is presumed to be the active site is selected as a grid. Parallelly, a compound library is selected and database of the compounds is made. And then computationally, this library of the compound is screened for binding to the active site. With the aim to reduce the time, the high throughput virtual screening is again divided into the 3 steps: HTVS, SP docking and XP docking. The major difference is that in the high extra precision docking, what docking of the one compound takes generally 3 to 5 minutes in a workstation. Why? In the standard precision, it takes only 30 to 50 seconds, and in this high throughput virtual screening HTVS, only 3 to 5 seconds. The major aim of this is reduction of the time, because all the compounds that are discarded that do not fit into the active site can be discarded into the earlier filtering step. Docking of the compounds into the active sites, that is in the cavity 2. All these compounds docks into that cavity. Further shows docking of the compounds into the cavity. in the active site cavity of the protein. The hits that are identified through the virtual screening are generally they bind tox to the selected cavity, the active site, but many times their binding affinity are not as good as required for our drug molecules. So to increase the potency of these molecules are further modified and docked into the same cavity, and their Ki values are predicted. In the another approach, that is ligand-based approach. Here, ligands where the 3D structure of the target is not known, the ligand-based approach can be used. And that approach uses a pharmacophore. Pharmacophore is what? Pharmacophore is the precise section of the molecule that is responsible for the biological activity. When a compound docks into the active site of a protein, all the atoms of that compound do not interact with the protein. There are only certain moieties that interact. Those interactions may be hydrogen bonding, they may be hydrophobic interactions, they may be van der Waals interactions, there may be water breeze, that may be salt breeze. And all those active compound moieties that interact with the protein and make it effective molecules to inhibit or activate the activity of that target protein are considered in the pharmacophore development. So students, let's now summarize what we have learned in this module. With the emergence of the new pathogen strains, development of the resistance to the existing drugs, now new drugs will be required. Second, with the availability of the genome sequences of the different hosts and pathogens, novel drug targets are being identified. And computer-aided drug design has considerably reduced the time and money required to identify new drugs. High-throughput virtual screening can screen millions of compounds within a span of the few weeks. Thank you.